Hello and welcome to our new BioFlux live demo from South San Francisco. I will be soon giving the helm to Dr. Brian Haynes here at the lab. Please note that if you have any questions, you can enter the questions into the chat window or into the question window with your interface. Dr. Haynes will actually answer the questions as he goes along or at the end of the seminar. Thank you for attending, and Dr. Haynes.
Thank you. In our continuing series on live demos, we decided to do something relating to cell adhesion, as it's a very popular application with BioFlux. Our goals today are to establish a monolayer of endothelial cells, initiate cell rolling and adhesion, set this up for transmigration, and then briefly evaluate the data using cell rolling software. So the protocol for forming a monolayer, it varies between cell type and the conditions that you're looking to pursue. But typically I found that this general guideline makes the most sense. You start by priming the plate from the inlet to the outlet with 60 microliters of fibronectin. This can be done at 1 dyne or 5 dynes. I usually do this a little slower just to give myself ample time. Then incubate this for an hour at room temperature to allow the fibronectin to bind onto the glass. Follow that by a rinse with media for about 15 minutes. Remove the media from both sides, and it's really important to remove as much as possible, one, in order to keep the levels balanced, but then on the outlet well where you go to add the cells, it's important that that volume be as consistent as possible among the wells to keep your loading even. And then we'll prime this, the Hubeck cells at roughly about 15 million cells per mil. And I know that it sounds like a large number, but when you're just working with 5 microliters of cells, it's actually not that bad. And that goes into the outlet. We'll incubate that for about an hour and then add media to it. Now, I'm just going to go through these steps with the cameras, but I have a monolayer that's formed from last night. And for these purposes, I'm just going to do one column here as a demonstration. I'm going to add 60 microliters right to the middle of the well, and trying my best to avoid not actually touching the bottom because it's glass, and at the same time making sure that I don't have an air bubble trapped underneath of this. Anytime you're working with microfluidics, air bubbles can become a limitation. So run this at 1 dyne for about 5 minutes here. And actually it should go through in about a minute and a half. If I run it at 2 dynes, it'll go through faster. The reason I don't suggest running it at 2 dynes is that if you walk away from it and with a lower volume, you're more likely to run through the material faster. There are a couple ways you can tell if the liquid's perfused through. You can put this on a microscope, but I typically hold it upside down. Let's see if I'm in the camera here. I see a bead of liquid forming on the other side, and that's sufficient to know that the liquid has gone through. And if I was doing this in real life, I would incubate this at room temperature for about an hour. Though most likely the protein binding activity happens within about 30 seconds. I'm going to rinse this off with media. For a while I was using a PBS, but when you think about both the blocking effect of media that has some sort of serum in it, and that the conditions in terms of what's healthier for the cells, media makes actually more sense than using PBS, or rinsing with PBS and then rinsing with media. So I put that media into the outlet well, and I'm going to run it 2 dynes for, if I was doing this in real life, for about 15 minutes. And we'll just run it to the point that media comes to the other side. When you have a phenol indicator in the media, usually you can see it pretty easily once it's gone through. So stop this. Typically the media would have gone farther, but again, we're just doing this for demonstration purposes. Usually I remove from the media side first, and then from the side with the fibronectin end. And if you take your pipette tip and you just go along the side of the well, and you rest it on the bottom, you don't need to worry about damaging the plate because you're just hitting the bottom of the plastic record titer plate. And using this does just a quick pass of removing the bulk of the media from this side. And then typically I remove from the inlet side as well. And usually after about 15 minutes you'll have about 60, 70 microliters to pull out, minus the remaining material. And then I'll follow up with a secondary aspiration. If you use a vacuum pump, you can do this all in one step. I like the control of having a pipette, but some people prefer the vacuum pump. And when I remove the second amount, I rest my tip on the PDMS layer, which is the middle ring, and then I aspirate, leaving a bubble of media in the very center, and everything else being aspirated out. The next step is going to add these cells. One of the biggest problems that people have when forming a monolayer is that they need to have concentrated cells, and they need to be healthy. And the cells are healthy and concentrated, the monolayer will actually form within about 5 minutes. The other thing to consider is that when you add the cells, I like to move as quickly as possible, because when you work with cells of that concentration, it's very likely that they'll want to stick to themselves in the well that you put them, which will prevent you from getting them into the channel. So I like to work as quickly as possible, putting the cells into the well and then perfusing them through. And I'm going to do this for about 5 seconds at 2 dynes. Set this up for 2 dynes from the outlet. And count for 5 seconds. A little tapping. Not really sure if I have data that says tapping helps or tapping doesn't help, but it definitely makes me feel better in thinking that the cells are agitated and moving from one side to the other. We're going to take a look at this with our imaging software called Montage. And I've got this in bright field, so I can lower the exposure a little bit, and you can see the seeding consistency. Doing this with slightly fewer cells will be fine. It's just a matter of how consistent you are among your monolayers so that you have consistent data. One of the nice things about HUVECs is that they actually spread quite dramatically. So when your monolayer is formed, you might be hesitant to think that it's completely coating the entire channel. But when you look at the this under with actin staining or some other methods of imaging, you'll see that the entire channel is actually filled up. So at this point, I would stick this into the incubator, standard tissue culture incubator with humidity and CO2, and let it incubate for about an hour. Some people like to go a little longer. Some people, if they're in a hurry, can go a little shorter. The real advantage to doing it, about an hour and a half, is that you know that the cells have adhered. The thing to keep in mind, though, is that if you leave it too long, it'll dehydrate out. And so after you've done that, what we typically do is we add media to the inlet, which allows gravity to feed the flow cell and to feed the cells. And then put it back in the incubator and leave it between 4 hours to overnight. Some research groups, like our one at MIT, like to start early in the morning. They'll see the cells, wait 4 hours, activate the cells, and then move on one step at a time. For us, we found that that's not really may not be practical, and so a little bit an alternative is to do this at the end of the day, and then come in the next morning, activate the cells, and then continue on. So to show you what a nice monolayer looks like. Now, if you look at this in bright field, you can see the outline of the cells. And actually, this is in bright field. Putting it in phase contrast, you can get a different dimension for the cells. And when you want to do transmigration experiments, for example, the cells that are rounded and at the top that have not adhered will look white. like the one or two that are here, and the cells that have made the monolayer will look darker. So the next step in doing this is to activate the monolayer. And so I have a brief slide about that. We're going to perfuse 200 microliters of, in this case, I've got 50 nanograms per milliliter of TNF alpha at 1 dyne for 5 minutes and allow the cells to incubate for 4 hours. People have asked, what does TNF do? And it's typically considered the gold standard or the classical standard for endothelial cell activation. And really, more of the question is, what doesn't TNF do? In many cases, it starts out, you've got phospho-AKT, and within about 4 hours, you've got other post-translational modifications affecting the integrins on the surface of the cell, allowing for adhesion. An alternative to this, which doesn't require activation, is to overexpress an integrin or an adhesion molecule on a standard cell line. An example of that, a group at Harvard's done this as well, is, for example, E-selectin on CHO cells. That's a slightly cleaner mechanistic study, though maybe not as relevant when TNF, you've got the full picture. When you're throwing on large amounts of E-selectin, for example, you're just looking at E-selectin-mediated adhesion. So I'm going to put the TNF alpha in the top row. And the media in the bottom. You could leave the media from the day before. I think that's a little bit cleaner to use fresh media if you're using fresh media with TNF alpha. After you've incubated the cells overnight, it's very common that an air bubble will form at the bottom of the inlet well where the media is. It's very simple to get that out. You just pipette up and down until the bubble goes away. If you don't do that and a bubble gets trapped into the fluidics, it might not destroy the monolayer, but it'll certainly affect your data, and it'll just make for a messy experiment. So it really only takes about 30 seconds for a whole plate, but it's important to get that bubble out. So we go to my control software. I'm going to run at 1 dyne and go from the inlet to the outlet here. We can stop that now. Now, in terms of the next step for this study, what we plan to do is perfuse over 200 microliters of adherent cells at a concentration of 3 million cells per mil at 1 dyne. Now, the shear rate can vary, and you can go from as low as maybe 0.1 dyne. In these plates, you can go up to 20 dynes, and we have some plates that will go up to 200 dynes. For these type of cell adhesion, cell rolling experiments, usually people go between 1 to 4 dynes. There are some groups that are really interested in adhesion at 20 dynes and think that P-selectin is mediating that. In that case, people have found success by priming with a lower rate and then creating a ramping program to ramp up the flow through. So we're just going to use a pair of channels here. And I have HL60 cells that have been labeled with calcium, so they'll come up green if you want to put them in fluorescence. And we'll put these on the scope and take a look. While 10x looks nice, we can see both channels with 5x. And we started carrying the Orca camera from Hamamatsu, and that enables us to have a larger viewing window. The advantage of that is in 10x you can see two channels clearly. I'm going to start here. So you can see things are moving, and pretty soon we'll see cells being perfused through. And we can switch this to FITSI. The top channel had TNF, and the bottom one did not. And you can see here that cells are sticking. If you want to do rolling studies, this needs to be done by streaming. Or you take a, how can I describe, as short of exposure time as possible. I can move that over just a little bit and you can see. The top one again are treated with TNF and the bottom one is not. To stream this, we go to MM Standard. This is with the Montage software. Acquire, stream acquisition, and then we would pick the wavelength, the frame number. I typically stream it to RAM and then hit acquire. If I were to do this, it would, aside from tying up the computer, you won't be able to see things as requiring. So I'm not going to acquire this particular image stack. But you can see the cells here are rolling. And this could be recorded either in fluorescence, or if you want, you could do this in bright field. Now, an alternative is to look at this in phase. And you can see the cells, the ones that have adhered, the ones that haven't. If you want to look at, the adhesion profiles, typically after you flush the cells through the system, it's good to rinse the cells with media, and that enables a clean flush, and that way there aren't cells that are just in the liquid that are being imaged. If you want to look at rolling, you need to do it while the cells are going through, or once the cells have adhered, you can clean it out with clean media and then continue to image. For transmigration, you need to set the cells up like this and then put it in a phase contrast similar to this, though you could do at a slightly higher magnification. So let's try 20x here. Need more exposure. In this case, you can see the cells that are on the top, and of course you can see the rolling profiles as well. But as they migrate through, they'll go from bright to dark, and that will enable you to gate it back and forth. In different phases, this is phase 2, and depending on the focal plane, will enable you to do the transmigration experiment. That will take approximately 6 to 10 hours in order to do that. So I got a question about how long you let the cells run through. This depends on your experiment. The biggest thing I tell people is that you need to be consistent among your cell types. But the short answer is that for these type of adhesion studies, 15 minutes should be sufficient with this concentration of cells. 15 minutes of running and about 5 minutes of rinsing. Other people have run longer periods of time, and of course for the transmigration, you do need to run them longer. These are HL60s. They're more likely to roll than to adhere. And of course, I'll show you briefly here on the side window. If I increase the speed of this, that the cells roll off faster. So now we're up to 4, 5 nines. And most of these have rolled off. HL60s are good for rolling studies. They're not so great for firm adhesion. And stop that. And I'll add some more cells to another channel to take a look at here. So these are THP1s, a monocyte line that can be aggravated with something like PMA or LPS to grab down. Typically, they're not adherent in the unactivated state. We can look at these at 5x. And we'll start with a bright field here. We'll lower the exposure and diffuse at 1 time. So this is the right half of the plate, channel 17 to 18. So we'll do the lower half here and run from here. Oh, I'm looking at the wrong channel. So again, the top channel has the TNF, and the bottom channel does not. Looking at this in FITSI, you can see that the cells are adherent, but they are rolling at some degree. They do stick a lot more than the HL60s do, for example. Gating this is quite easy in fluorescence because you can threshold out these circles versus anything else that's background. Now, while this is running, I'm going to go through a few slides here to explain a little bit about the BioFlux and why we're doing this. So again, to refresh, I perfused 200 microliters of adherent cells at 3 million cells per mil at one time. For rolling, images need to be streamed or as short of exposure as possible. For adhesion, it is better to rinse out the cells, though if you do it kinetically and you do it under fluorescence, then it's not entirely necessary. And then for transmigration, it's a good idea to rinse out the cells, though again, not entirely necessary. But you need to set up in a phase contrast system so that you can see the cells on top versus the cells below it. Now, looking at the mechanism of cell adhesion, I was in Korea a couple months ago and trying to explain this to a group of people. And eventually I had to get down to the simplest possible way and said, okay, if you look at an empty plate, you think of this as your BioFlux channel. And you can put on a monolayer of kimchi. And this will coat out the BioFlux channel. And the chili on the kimchi really serves as the effect of the TNF alpha. These are the integrants that are on it. Now, to follow up with that, you can either take tofu or roasted garlic or potatoes in some case, and they'll adhere to the surface of the cells. When you leave this longer, though, you will get transmigration. And so looking a little bit deeper into this, the classical mechanism for tethering and rolling adhesion typically revolves around E-selectin. And then for firm adhesion, people look at towards VCAM1 to also P-selectin as well. Though P-selectin has been shown in rolling adhesion just in high shear rolling adhesion, close to 20 dynes where it will grab on. And then the functions of transmigration typically revolve around ICAM and then also alpha 5 beta 3. So again, just to explain, this is the monolayer cells. These are the cells that will be floating through circulation, and they would start out with a tethering, rolling phenotype, moving to a firm adhesion, and then to transmigration. Different ways that people have studied this type of thing. Laminar flow cells have been quite popular, and they worked really well. It's just the throughput is severely limited, and the amount of media that it takes to do this is also a problem. The way the BioFlux system works is that it allows for a pneumatic source in a physiologically relevant way. You can control the shear. You can modify the program to push the shear in one direction or the other. You can control the gas that goes into it, temperature control, and other variables. The typical disadvantage to this is that you're limited to about 24 assays, but actually you can multiplex this and do 96 at a time. We have several industrial customers that are using this and running 96 assays at a time, or 4 plates in parallel. And for the adhesion studies, this is very practical because after adhering the cells, rinsing the cells, and then you go to image the cells, you'll have 4 plates completed at one time. And so in comparison to approaches, for throughput, the static weld plate does give you high throughput, but it's really not relative, and a lot of people struggle doing adhesion studies with static weld plates because you might try to rotate them or rock them to stimulate how the cells will be interacting with a monolayer, but it's quite difficult. And flow cells are very physiologically relevant, They're just they're limited in the throughput. And we like to think that the BioFlux sort of combines both of these. The BioFlux 200 system controls shear. It allows the channels to go on and maintains temperature. It includes the BioFlux plates and then the basic software. The 1000 system, which is what we're looking at here, is includes a Zeiss microscope, an automated Zeiss microscope, montage software which controls the microscope and then also controls different functions for analysis, and then automated stage and automated fluorescence. The way the channels work, we've got two basic types of plates. One is a 48-well plate that allows for one input and one output, the liquid to go from one side to the other, and then cells would be imaged here, which we've been looking at. The 24-well plate allows for two inputs and one output. And an example for cell adhesion is that if you didn't want to stop the interface and rinse it out, you could put your cells on one side and your rinse on the other, and perfuse the cells and then perfuse your rinsing media. I'm sorry, I'm reading one question here. So I had a question about whether or not we can use collagen. And that's doable. We have a webinar that was recorded a few months ago that looks at collagen and whole blood, and invites you to take a look at that. In terms of, can you look at B-cell homing into the basement membrane? That's completely doable. We have a lot of publications that have come out about that, and I'd be happy to follow up with you. So applications for cell adhesion include inflammation, and I guess the past 15 years, people really focused on expression, gene expression, protein expression, what the inflammatory system was doing. But if you look at the basic mechanism of a circulating leukocyte or lymphocyte to be able to adhere onto the vessel wall, if that mechanism is compromised or overactivated, that can have serious consequences both for fighting inflammation or suppressing inflammation, such as in vascular diseases and stroke and heart attack. Another really big example is cancer. That primary tumors need to shed effective circulating tumor cells into the system. And what I mean by effective is that the primary tumor sheds cells into the circulation, and they come back and they're EBCAM positive and they're cytokeratin positive. That's great, but if they need to be able to adhere onto the vascular endothelium in order to spread. And so one of the things that makes a circulating tumor cell effective or nasty is that its ability to adhere onto on the surface. And we have quite a few publications that have come out about that. And then another one that we have a lot of researchers working on is stem cell therapy. It's not something you would think of initially, but if you get a perfusion of stem cells into your circulation, if they're not delivered locally, like for example to the spinal column, but are put into circulation, they need to be able to home in on a place and then to transmigrate through. So all three of these are great applications, and actually there's quite a few more. Now, some of the immunology papers that have come out wind up in JI and some other articles that look at migration and adhesion. And actually online on our support site we have quite a few more. I'm going to switch to a brief movie, but first we'll take a look at this this case. So this is two channels that are flowing at the same time. They both have THP1s. The top channel of Huvex was treated with TNF-alpha about 4 hours ago, and the bottom channel was not. And we can take a closer look at that with a higher magnification here. Probably of the channel that has the cells and not the channel that doesn't. So while we have several cells that have adhered, we have quite a few that are also rolling in process here. And we don't need as much exposure at the higher magnification. Now, some of these cells appear like they want to begin to migrate through, but there's several others that are rolling along, like in the first step of rolling, adhesion, and then transmigration. And then on the bottom channel, you may see some streaking. However, none of these cells are actually adhering. And if you want to increase the flow rate, you won't see anything at all. They blow past. But the big takeaway message here is that the cells that are treated with TNF-alpha are adherent, and the ones without it are not. Some people look to block this specifically. Other people look at, for example, the effect of diesel exhaust on activating cells, like TNF-alpha would. I'll go ahead and pause this. With the remaining time, I'm going to show you briefly here a little bit about the analysis. Though to go fully into this, we really need another webinar. And so we have a video here of platelets rolling over a VWF subunit. Whole blood was perfused over VWF, but the cells were rinsed, and adhesion was studied at 40 dynes. And at 40x. So these are our platelets. They're pushing along, and the software. So we've got two different software options. One is on the montage, which is the multidimensional motion analysis, and the other one is something called bQuanti, which was from one of our customers in Italy that wanted something that was more relative to their research with cell adhesion and cell rolling. I have a quick question here. How can I coat gelatin in the chamber and grow Huvex? Will gelatin block the chamber? So if you want to put gelatin in the chamber, I suggest doing it at between 0.1 to 1%. Though I have to say, my experience, fibronectin works much better. And if it starts to slow down the chamber, you just heat it up to 37 degrees. And then I have another question about transfected stem cells. There's at least 2 publications on our website about transfected stem cells, mesenchymal stem cells and drug-treated stem cells. And looking at them, of course, there's always a concern that you've damaged the stem cell at some point. But if you haven't sufficiently damaged the cells and they're healthy, they should adhere if they're going to adhere. So with the bQuanti, I have some screenshots to show you how the analysis would look. The program will pick up the cells. The only thing about this particular screenshot is that this wasn't calibrated to the size that we were looking at. This was a 40x magnification, so these sizes are slightly off. But the flow is going from the left to the right, and depending on the arrest threshold and the sub-rolling threshold, those values are set. And then tracking takes place using the software, and you can see the cells that have arrested that we're saying between 1.6 seconds and 2 seconds, 2 to 4 seconds, and then cells that were rolling and cells that were considered tethering. Then the morphology takes a stab at showing, the cells that appear to be fully round, the ones that look polarized here, and ones that just don't really fit either phenotype. And then looking at the events, you see that you've got about 45 cells that were tethering. Actually, the total number of cells that was recorded on this was 1,272. Most of these were rolling, and then different levels of arrested cell movement. And then looking at morphology, most of these were round. A good portion of these were polarized, and then some were just unclassified. We are pushing 6 o'clock, and I'm at the conclusion to this. I'm happy to take any questions and invite you to a follow-up in about a month and a half. We'll send an announcement for a more in-depth analysis of cell rolling.